So today, I'm going to give two talks. So this is going to be like the teaching talk, and then in the afternoon, sort of the research talk that will maybe wrap everything up, maybe. So last time, I introduced the basic one locus model that underlies sort of this idea of a multifactorial model for quantitative genetic traits. And today, we're going to talk a little bit about selection on quantitative traits and fitness. And we have been talking about fitness all week. And so we have had several definitions of fitness, all are fun. So I think Daniel sometimes defined fitness that sort of we have a birth rate minus a death rate, right? That can give you a measure of fitness. The first lecture I gave, remember we had haploid, two haploid strains, right? Two haploid strains, and one was growing at rate little a, and the other one was growing at rate little b, and we defined a measure of fitness as sort of the relative ratio. We used that as well. And we've had a whole bunch of different measures of fitness, so today we'll sort of continue thinking about that. And when I teach this to my students in the US, I always draw this picture, and I promised them I would draw it for you guys too. So if we imagine a life cycle of, and we like to think of this as an arbitrary population genetics organism. So you might imagine there are eggs, or larvae, something. Some of them are different sizes apparently too. And then they might grow into some, it has to have a smiling face. Some intermediate, so larval form. You might imagine that then we have adults. They also smile. Somehow they have 4 legs at this point. And then, they have eggs. So we draw this because it's completely not real, and so it's more fun. So if we think about fitness, we have to sort of define a beginning and an end. And oftentimes we say, well, given, you start at one point in the life history, how many eggs does an egg end up leaving in the next generation or something like that? But that means that you can also define components of fitness. So you could have survivorship from one stage to another, and you could have fecundity. And so another definition of fitness that we did in the age-structured and stage-structured cases were, we had the growth rate, the leading eigenvalue of the matrix, the transition matrix, which, in the age-structured case had fecundities in the top row and survivorships in the subdiagonal. And so the leading eigenvalue was using information from the life history, but in the same way, to measure the amount of growth or how many of an organism it leaves in the next generation. So that's another definition of fitness. We use, and sometimes also we did, logarithm of that. But all of these have this relationship that we're looking at sort of how things sort of replace themselves, and they can be relative or absolute, right? Okay. So just sort of to remind us that we've been thinking about fitness all along. And generally, so for today's lecture, I'm going to think about survival as a measure of fitness. For right now. And we might say, well, fitness is 1 if you survive, and fitness is 0 if you die. So, and then we might say, well, what if we decide that fitness itself is the quantitative trait that we're interested in? And that makes sense because we imagine that fitness is actually composed of many, many sub-things, all of which probably have many genetic and environmental inputs. And so it makes sense to treat it like the type of trait we were talking about before. And so if we have a population of size n at some stage, maybe here, and we say, well, for each individual in that population, there's some fitness, Wi, which is either 1 or 0. Then we can define a mean fitness. Right? It's just the mean of those. And we can define a variance in fitness. It's the variance in those 1s and 0s. And because fitness, we're going to say that fitness is a quantitative trait, like the traits we were talking about last time, in principle, we can calculate all the components of the variance. So now this is the same as before, but before I was using an arbitrary trait, and now I'm going to use a little W to indicate that I'm talking about fitness. So we might have the overall variation in fitness, right, which is, calculated the way we always do a variance. Right? So this is actually the variance in these ones and zeros. And so that's what this is. And then we say, well, but really we can decompose it the same way we did last time into a part that's the additive component, a part that's the dominance component, an interactive component that's due to any epistatic effects, any sort of independent sort of environmental effects, and then all the possible covariance terms that we could think of. And then we can make different kinds of assumptions to maybe say that those covariance terms are zero, or we can, in cases of an experimental population, we can set up our breeding program so that some of the covariances with environment are zero by doing common garden or by doing randomized environments and things like that. So when we do this, when we have this breakdown, decomposition of the variance in fitness, it lets us write something that you've already seen before in terms of this, which is Fisher's fundamental theorem. And once again, no one knows why this is fundamental and nothing else is fundamental in evolutionary, but I don't think he named it that, so. Well, he was, I'm sure. He was famous for being quite not shy, I guess is the word we would. So one of the many ways to write this, Fisher actually did not define it exactly like this, but we're going to, in a little bit, derive this, that the change in mean fitness is just given by the ratio. Well, not the ratio, but the scaled, if you like, the scaled additive genetic variance in fitness. So this component of the decomposition. So we're going to come back to that. So let's think of a really simple situation where I have a population of size n, and I take some group of that population and they die, and some group of that population live. So it's very simple. So we have some large number n of individuals. We have n sub s, say, is the number of survivors with fitness 1. And then obviously n minus ns are the ones who die, the dead. And they have, 0. Then obviously we can, because the mean fitness is just the sum over the w's, the mean fitness for this will just be the ratio, right, of the survivors to the total population size. It's the proportion of the survivors. So this is a very simple case that lets us have something simple. So now let's think about a phenotypic trait that we're interested in these organisms. And it could be anything. It could be the size of the organism. It could be things like the day of first flower, if you're talking about plants. It could be the number of eggs laid or anything you think is associated with fitness. So we have a phenotypic trait we'll call X. And that trait also has a mean and, a variance that you can measure in the population. And we can also, given that some individuals survive and some individuals don't survive, we can measure the change in that trait. And we've defined that earlier. It's the selection differential. So earlier I think I don't remember what I used, but now we're going to do this. It's just the difference in the mean of those who survive for this trait and the mean of the population as a whole. I think earlier we might have written it as minus m or mu. I've been very inconsistent with my notation. So x bar s is just the mean in the n sub s survivors of this trait, whatever this trait is, maybe the size or anything else. And we can rewrite the equation for the mean of the trait and the survivors by just weighing the x1s by 1 if they survive and 0 if they don't, so the fitnesses. So this is also this sum, you know, each xi being weighed by the wi, right? And a little bit of algebra leads us to write this as this quantity. Because we're using the fact that W bar is an S over N. So we can write the selection differential as this quantity. Which, with a little bit of moving things around and assuming N is reasonably large, gives you the covariance of X and Y. X and W, sorry. Not using Y. In terms of W bar. And sometimes that's written this way instead, pulling the W bar inside. So sometimes you write it like this. So it's the fitness relative to W bar. So this is just saying something sort of obvious, that when you have selection, especially the simple form, but it's generally true, that the strength of selection gives you the covariance between the trait and sort of this fitness, but whether it's going to survive or not. So then we can go back to the Breeder's equation again. So, you know, we had this square there. The response to selection is just the heritability times the selection differential. But we have previously defined the additive genetic variance over the phenotypic variance. I'm going to put an X now to indicate it's for trait X. So that's H squared. And then we can write here this covariance. Like that. There's a few different ways to write that, but we can also do this. I want it in a particular form. These two things. And this is the regression of scaled fitness onto the trait. So giving myself a little bit more room. So if I have sort of scaled fitness here, and I have the trait X, this is, in this particular example, I just have two different fitnesses, 0 and 1, right? Maybe I imagine that for this trait, individuals with higher values of the trait are more likely to survive. Maybe they're bigger or something like that. We might see something like this, data that looks like this. And, you know, here at 1, some distribution that looks like that, and if I fit something to this. Right? And obviously it could go down, or there could be no relationship. So what this is doing is writing the response to selection in a little bit different way, yet again. We're writing it as a part that is, you know, sort of purely genetic, if you like, that's purely part of the genotype, this additive genetic variance that we've been talking about, and then a part that is really a description of phenotype, because this is just a description of how phenotype and fitness are related to each other. So we've decomposed the response to selection to these two parts, and that's basically what the Breeder's equation does, and so often you'll see it written this way. And later, at the end of today's lecture, we'll just generalize that for a multivariate trait that has more, where we're following more than one trait at the same time. But that's sort of a general result. So I promised that I'd derive Fisher's theorem. And this is like the simplest derivation in the history of man. What if the trait, X, is fitness itself? So we ask, what is the response of fitness itself? So in that case, R is the response, is, you know, the new fitness, and the old fitness, we'll call that delta W bar. And using this, it must be the additive genetic variance for fitness times this covariance, the covariance of fitness to itself over the variance in fitness. And just a little bit of rearranging gives you Fisher's theorem. Because obviously the covariance of fitness itself is the variance of fitness. So the things cancel. So this is one way, using this sort of quantitative genetics framework to spell out Fisher's theorem. What's it saying? Why is it fundamental? It's basically, I don't know why it's fundamental. Somebody named it that, but it's saying that as long as there is additive genetic variance for fitness, you'll see a change in fitness. So there may be other kinds of variance in fitness, right? We might have dominance variance, we might have epistatic variance, other kinds of variance or covariances. But if there's no additive genetic variance in fitness, then we're not going to see a change in fitness. So that's the sort of the thinking there. Yes? I'm so wanting you guys to do an exercise, but so I'm going to maybe we'll do it. So in the handout, there's a toy exercise that is probably done in every population genetics class in the history of population genetics. Maybe. So it's this idea that you observe an event that kills off part of a population. We were talking, I forget what we were talking about, this idea of like, maybe there's a cold frost or something like that, sort of a selective event. And then you could ask yourself, if you observe such an event, what if you had a trait you were interested in, and you measured the value of that trait before and after selection? Classic example of that sort of thing is the study of the Galapagos finches, where they look from year to year at the survivorship of the finches in a dry year or a wet year, and look at traits like the beak length or the beak depth, and ask the question, you know, do we think there's selection? Could selection be acting on this? Is there a selection effect? So in the toy example that's in the homework, which we're going to go through, because I don't believe that, I feel like we should do at least one. Let's say we have some birds, and there's a storm, very bad storm. And there's a building on campus at Kansas that has these big windows, and every time there's a storm, birds get thrown against the window and they die. Right? It's very sad. But as a biologist, you're like, aha, this is a selective event. Let's pretend, this is not true, that all the birds were all the same, from the same population. So there are a group of sparrows and all the same population. And so you, being an enterprising young scientist visiting me in Kansas, go out with a bag and pick up all the birds. And some of the birds are dead, and some of the birds are just stunned, you know, because they hit the window. And so what you're going to do is you're going to measure something. You're going to measure their wing length in centimeters. You know, no one's going to stop you. No, people are picking up dead birds. Pretend that no one's stopping you. And you're going to measure the wing length and the ones that survived and the ones that died. So there's a, right? So this is your data. And maybe, thank goodness, there's a slightly smaller number of ones who die. So you look at this and you say, well, you know, if this was truly a single population, you know, what could we say about selection on the length of the wings? So what you could ask is, how does the mean in wing length change from one generation to another? And you have this exact same situation where you just have two fitness measures, 0 and 1, and you're basically, you know, graphing that relationship. So, you know, you would find the means, the mean fitness in this case. So all of these little fellows have a fitness of 1, and all of these little fellows have a fitness of 0. They're all dead. The mean fitness is just, the proportion, which happens to be 8 out of, is that true? I cannot be right. I have 8 out of 14. It is. Okay. I doubt my own math. Never do simple addition in front of an audience. Never. It's a bad idea. Okay, so about, a little over half survive. And you can calculate variance and fitness, same thing. I did this on a calculator, so it's probably a little bit wrong. And then you can calculate the regression coefficient of fitness and wing length exactly, sort of, you could plot this if you wanted. This ends up being, you have to calculate this covariance. It ends up being 0.2363, and a little bit of math gives you 0.811. Okay. Depending on how you do it, you could get quite a bit of variation. These are small numbers, so how many significant digits you have. So this gives you the relationship between wing length and survivorship. So what is it telling you? It's telling you it's positive. So it's telling me that the birds with the bigger wings were more likely to survive. I don't know why that would be true, but it is in this example. So now I have a question for you. What else do I need to know what is going to happen to my population of sparrows in Kansas that have survived this wing? So if I want to know, is wing length going to increase, or what other piece of information, what do I not know? You guys know it. You're saying it softly. We need to know something about the heritability, right? The additive genetic variance. Because we know that there is a relationship between fitness and the phenotype. We've established that in this very ridiculous example, but it still works. But what we don't know is there a relationship, an additive genetic relationship between the phenotype, underlying the phenotype? We need to know that. So how could we, if we were experimental biologists, how could we do this? There's 2 different ways, right? You could develop a breeding colony of sparrows and look at the response to selection in a controlled experiment. It's a little crazy. Or you could use the resemblance between relatives. If you could measure the wing length in a group of parents and offspring, remember, that gave you, if you use just one parent, the relationship between the two will give you twice that gives you an estimate of heritability. So you could do it that way. But you need more information, right, to understand if selection could actually do anything. So whenever you see, and so this is a silly example, but people do non-silly sorts of, where they go out and measure from year to year. So you could think of a year as being a selective event. If you have organisms that reproduce each year, the being able to survive and reproduce is the selective event. And then you could look at year to year how a phenotype changes, and that tells you something about the ability or the potential for there to be selective change in the trait. And people do it for all kinds of interesting traits. Okay, well that seems so nice, so simple, so easy. Of course, it must not be true, which is not, because what must be true of, wing length in sparrows. If bigger wings are better, then why don't sparrows have really, really, really long wings, right? There's obviously more to it than that. And one of the additional things is the fact that traits are correlated. So they're not independent. And so we're going to briefly talk about what's called the correlated response to selection. I've learned my lesson about the water. So imagine we have now 2 traits, the next possible iteration of complexity, right? So we have a group of individuals, and then we have trait X and trait Y, and we measure values of those traits in all of our individuals. And obviously we can, for each trait independently, we can calculate the mean, variance. we can ask questions about what the distribution of those are. And then we can also ask, how do those two traits covary? So again, a phenotypic, just an observational thing. A phenotypic covariance, right? Which is written many ways. We have so many things we have to keep track of, right? And that's just the covariance of those two traits. And we could summarize the relationship so that we can compare different types of traits by looking at the phenotypic correlation coefficient. Which I'm going to write with a subscript P. And it's just that covariance, you know, standardized by the standard deviation, so it lies between 1 and -1. So as long as this correlation coefficient differs from 0, there's some correlation. It could be positive, it could be negative. We can think of all kinds of traits that are correlated positively, like general traits that have to do with size, and traits that are correlated negatively. You can imagine those as well. We'll think about that, but we also are interested in genetic covariance, and we're especially interested in the additive genetic covariance, because the additive genetic covariance tells us the response of trait Y to selection on trait X, or vice versa, the response in trait X to selection on trait Y. If there's no additive genetic covariance between the two traits, then selection operates on them independently, changing one without affecting the other. But it's rarely true. So if you direct selection on X, gives you a response in X, which we've written various ways, but I want to write it like this. In terms of the selection differential and the additive genetic covariance. But if I directly select on X and then ask, what's the correlated response in Y? I find, I'll call it correlated response in Y, that it's governed by the additive covariance between X and Y. And a little bit of rearranging gives this version. I wanted to write it this way because we can say, okay, we have a response that's direct, and we have a correlated response in a different trait, indirect. And you see that it has this very nice and beautiful, simple structure, right? So you apply selection on X, and the response on X or on any other trait is governed by either the variance or the covariance of additive genetic traits. And often we summarize those in a matrix of additive genetic variances and covariances. I'll draw one at the end when I briefly talk about that. And so this is what's sometimes called the correlated response. It's also in the literature called the indirect response. And obviously, if this is true for selection on X, it's also true of selection on Y. So if we have direct selection on Y, the response in Y is given by this, and then the correlated response in X is given by the same quantities with additive genetic covariance. The response in X and Y, so it goes in both ways. And in the same way that we defined a correlation coefficient for phenotype, we can define an additive genetic correlation coefficient these are the square roots of the additive genetic covariances. So this allows a sort of a discussion of how these correlations might be related to each other that is related to what Deepa was talking about in her research seminar, where you're looking for correlations between traits because it keeps evolution from being simple, right? You're looking for things like evidence of antagonistic pleiotropy and evidence that things are not simple. So I don't think it necessarily might surprise you that the additive genetic correlation coefficient and the phenotypic correlation coefficient don't have to be of the same magnitude. They don't even have to be of the same sign. And I'll give you some examples in a little bit. But you know, they're free to vary. And so the question is, well, how are they related to one another? Obviously, they must be related to each other in terms of these variances and covariances and things that we've been defining because they come out of the same model. So what's the relationship between the two of them? We can define another correlation. It's a little weird, but it's called the environmental correlation. This is one of those things where the name is deceptive. So we'll maybe write it. So the environmental correlation coefficient. Now, strictly speaking, we can define correlation coefficients between all of those, I've erased them, but all of those components of the model, right? The additive and the dominance and the interactive and all these things. But we would like to separate the correlation sort of between two things. Genetic correlation that's additive, because we know that's what's important for the response to selection, and kind of everything else. So we're going to define, and here I'm following the notation that was used quite a long time ago by Falconer. So other people use different notation, but this is what I learned when I was young, so I keep using it. So we define a quantity E squared, which is just 1 minus the heritability. So obviously, if the heritability is the additive genetic variance over the phenotypic variance, this quantity is sort of all the other things. Over the phenotypic variance. And if there's covariance terms, they would pop into that as well, right? And so it's saying that it's the proportion of the phenotypic variance that is not the additive genetic variance. And a silly amount of sort of rearranging of algebra can show, once we've defined this, then the relationship between the phenotypic correlation and the additive genetic correlation can be written this way. Just for two traits. I mean, you can extend it arbitrarily for many traits. So these are the heritabilities for each of the traits separately. This is this environmental correlation. And I have to define one more thing. Well, we'll say that the covariance in phenotype between X and Y, we're going to decompose that into two covariances, the additive and sort of the kitchen sink, everybody else. And then the environmental correlation is just going to be the covariance over the standard definition. Okay, so what does this tell us if we decide to take on this? It says that that there is a relationship. Obviously, there's a relationship between the phenotypic correlation and the additive genetic correlation, but depending on the sign and the magnitude here, that relationship could be positive, it could be negative, it could be anything. So when we actually go out in nature, or sometimes not in nature, sometimes in experimental plots and farms and things, and measure these correlations between real traits, we see all sorts of things. And why do we even care? Well, if we have positive correlations, that must mean that there's some underlying factor that affects both traits that we're looking at in a similar way. And we can think of what's a really obvious thing that leads to a positive phenotypic correlation is environment. So probably the easiest thing to think about is something like nutrition, resources. So if you have traits that depend on the availability of resources for the organism, such as the size or the total litter it can make or something like that, if it's in a rich environment with lots of resources, those things are going to increase. And if it's in a poor environment with very few resources, they're going to decrease. And it might be purely environmental, and you'd see this positive correlation. We're especially, positive additive genetic correlation. This is one of those words I spell wrong all the time, pleiotropy. So if you have genes that commonly are affecting both traits, then you often will see a positive correlation. We're interested often in negative correlations. They keep things from being simple, and I'll give an example later, but they imply trade-offs. And the example of antagonistic pleiotropy gives you negative additive genetic correlation, is especially interested because it's sort of at the core of a lot of this theory on the evolution of life histories, where we're assuming that the total life history of the organism can't be maximized. We say, well, we should see negative correlations between it. And as we saw, we don't always see them phenotypically, and we don't always see them additively. I copied a few things from a summary, just because I wanted to, once in a while, it's nice to think of real creatures. I say this having never really worked on any real organisms of any kind. Yes? So, yeah, I can. Do you mean of actual values or? I don't know if there's a typical situation, but where people have looked, they have found values for all of these things. So I have a few examples. So these are from, well, one of them is from poultry, chickens. So here's X, here's Y, RP, RA, and RE. And so body weight and individual egg weight. And I don't think you'd be too surprised to find that these are positively correlated phenotypically. When you do breeding experiments, you can show that the additive genetic correlation is strong. And they also have some environmental. But another correlation with a body weight with, because you might want to have big chickens but also many eggs. Ha! No. So, there's some sort of weak phenotypic positive correlation, but you have a negative additive and sort of weakly. And there's no error on these. I ignored, there's error on the estimates of all of these. Obviously, these are estimated empirically. So, I'm not saying that these are the absolute values, but this is the kind of thing that people who study life history are always very interested in. This is a negative genetic correlation between two fitness traits, right? Because body weight might be related to survivorship, and eggs is definitely related to fertility. And you see this trade-off. This is a really common trade-off, size versus number of eggs. And then not, I guess, not surprisingly, other things. But so Drosophila, they've done millions of these. So bristle number in two different segments. So in the abdomen, abdominal, and sternopleural, which is another segment. These show positive And, and all sort of size type things almost always show positive. So in mouse, body weight and tail length is, generally related to total body length as well. And I think this was 0.45 and 0.29 and 0.56. These are bounded by definition between -1 and 1, so it gives you a sense of relative. So you can see the values are all over the place. Sometimes they're really small. This is a 4. There's many others. I just picked a few of things I know what they are. Some of them are really small, and that's probably not significantly different from 0, depending on the sample sizes that they use to calculate these, which I don't have. I checked this. This is significantly different from 0. That's a significant negative genetic correlation. And that's actually been seen in breeders who try to, you try to get bigger chickens and more eggs. You can't get bigger chickens and more eggs. You either get bigger chickens or you get more eggs, because there's a negative correlation of some kind between those two. Why does it arise? Well, so there's two sorts of explanations. There's the sort of vague, loosey-goosey one, which I have no trouble with that, but the idea of sort of a limited energy budget in the development of the organism, and you can either put in energy into becoming a bigger, fatter chicken, or you can put in energy into having more eggs. And that was the classic explanation for, the classic experiments on Drosophila, early fecundity as a trade-off to later for survivorship, putting energy into each. But that's kind of this loosey-goosey explanation. We really would love to know something like, what's the genetic? There has to be a genetic underpinning for that. So, I don't know anything whatsoever about the development of chickens or even birds at all, but we can easily imagine that, that there might be developmental trade-offs between large size and later fertility. But basically, to know the explanation, you need to know something about the development of those traits, right? Well, how are those traits? This is genetic, not environmental, so we need to know something about the genetics of those traits. For some traits, they have actually, I'm sure in yeast, we actually probably have the actual genes underlying some of these trade-offs, perhaps. Yes. Ophelia, please. Yes. Mm-hmm. Yeah. And they're the ones that respond when you actually do this sort of thing. Yeah. Yeah. And that's a really good point. This is, these are measured at a point in time. So this is what that doesn't mean this is what they were along the evolutionary trajectory. That's what the variation is in the population right now. And that's depends on the history of those populations. So yeah. And there's other explanations that I've seen in the literature, but I don't know that I believe one or the other. But, so that's looking back retrospectively. It does tell you, looking forward into the future, what's possible. So it's not clear that we can sort of just look at these and say anything about the past, other than we got to this point. But it does tell you something about where you can go from here, which we'll talk about in a second. Plenty of time. I have another activity, but I'm feeling incredibly lazy. But you will do it on your own, yes? This weekend. That's what you're going to do. You're going to do all the homework. Yeah. Okay. So actually, this leads. Ophelia sort of set the stage for the next thing. So we have these. This is basically description. We're describing the population right now, and then we want to. Our question is what happens in the future. And we know, because I already told you, that the variance covariance, that these quantities tell us the response to selection moving forward. So it's very easy to take what we've just done and go ahead and expand it to a trait that is multivariate. We might imagine fitness itself is multivariate, but almost everything, the phenotype of the organism. So this is sort of the what happens to the whole phenotype? Yeah, yes, no, more. So yes, generally. Yes, because remember, we don't have it up, but from last lecture, we wrote with some of these, even in our very simple toy model, these variances and covariances depend on allele frequencies. And so they're changing, and they depend on the phenotypic variance, which is changing, right? So they're changing. The idea is if there is enough, if they're truly quantitative traits that are not controlled by just a few loci and a few things, then the change should be gradual. And when people actually do selection experiments, you can do predictions using these for a few generations and sometimes more. You certainly can get a response to selection for very long periods of time. In some experiments, we were talking after class about the famous oil content in maize seeds, and that's been going on for decades and hundreds of generations. But it's almost certain that additional variation is entering that population through mutation because it's a very large population. So that's a whole question of what maintains the variation as you go. In general, the assumption is that the allele frequencies are changing relatively slowly and that the whole phenotype is changing relatively slowly. So it's approximately constant over short periods of time. So it predicts each generation what happens in the next generation. But you're right. These are a variable because remember we wrote these out and this is a function of allele frequencies. So it has to be changing if selection is changing allele frequencies. So if we select on several phenotypic characters at the same time, or if the character that we're interested is multivariate in some way, we might imagine this. That, not a bar, a vector. That Z is a vector of a set of phenotypic traits, right? And we want to ask the question, I'm going to apply selection to this composite, and I want to see what happens. You know, I sort of wrote it arbitrarily, and then I immediately am going to go to the case of 3, because that's, you know, the one I can write simply. So imagine that, that Z is, you know, Z1, Z2, and Z3. Well, if we look at these two equations that I have here, right, it's immediately sort of obvious what we can do. We can say, well, we're going to define phenotypic variances and covariances. So we'll have a matrix P, which gives in the diagonal the phenotypic variances. My notation. Now I'm going to have all horrible notation. Okay. Wishing I'd done something different, but too late. So we have all these covariances, and these are the phenotypic covariances. 1, Z2, and these repeat. 1, Z3, Z2, Z3, right? So we have all these, but obviously we also have, we can define a sort of sister matrix to this, whose entries are the additive genetic variances and the additive genetic covariance. And now I am going to be lazy and just write a couple of them. So this is the additive genetic variance here, and then you have the covariance terms, the additive covariance terms. Z1, Z2, covariance, Z2 additive. And in the diagonal we have, you know, these guys. And in the off diagonal are all the covariances, right? And that makes sense. And so in the same way that we wrote this kind of equation, the correlated response in Y is the additive genetic covariance between X and Y. X. We can say the overall response in Z, which includes both direct responses and indirect responses. Also write this way. And these are matrices, G and beta. Earlier, for a single trait, we had, the response in X, or the response in Z, so was Z bar prime minus maybe Z bar, something like that. So, and we had that the change in Z bar was this additive genetic variance times beta. Now we have that the change in Z bar is the matrix of additive genetic variances and covariances, and a vector, where that vector is just the vector of all the selection gradients and all the individual traits. These things. And that's the selection gradients. And each one of them, is just the covariance of trait Z, fitness, mean fitness, over the variance in Z, right? So it's that gradient of fitness with respect to whichever the trait Z is. And the other thing that you might note from this writing, this thing here, is a vector of the selection differentials, and each one of them is this sum. I think it's useful to look at. So for our 3 case, it's B1, B11, I'm sorry, beta 2, B12, beta 3, B13, which is beta 1 times the phenotypic variance in Z plus beta 2 times the covariance of Z1 with Z2 plus beta 3 times the covariance of Z1 with Z3, and similarly for the other ones. And so you can see that for each entry, for each trait, the amount of selection you're applying is made up of some direct selection that's mediated by the variation in that trait, and then indirect selection on the other 2 traits that's influencing that trait. So how much selection is being applied to trait Z1? Well, it has to do with how much direct selection is being applied to trait Z1, and then how much indirect selection is being applied to trait Z1 via selection on Z2 and Z3. And that's true for all of them. So for all of them, there'll be some amount of direct selection and some amount of indirect selection. And obviously, if for whatever reason there's no covariance, right? If there's no covariance between the traits, then these matrices are just diagonal matrices, and these terms go away, and you would just have direct selection. So why is this interesting? I mentioned this before. It's interesting because it keeps selection from being simple. Now I'm going to draw very poor fitness surfaces. I feel bad because I feel like you've seen some very nice drawings, some very nice sketches, and it's not what you're going to see now. Beta is basically it's a slope of how the mean fitness changes with sort of mean phenotype for a particular phenotype. So, for a single character, we can draw sort of simple case where we have mean fitness as a function of x. I've drawn it the way everyone draws it, where there's a single peak, just because that seems reasonable. And then, the slope at any given point gives us the relationship between fitness and that trait. And for two characters, right, we can easily draw, well, I don't know, easily, but this is one of those, in 3 dimensions. So here's x, here's y. Something like this, and we might have some sort of, I don't know, 3-dimensional thing. It could be anything. It changes. Let's imagine that we have just 2 traits, just for a second, and we have some sort of simple part, at least, of the fitness landscape where we're focusing on at least, even if it's not the global maximum, a local maximum. And we're going to draw sort of a contour. And we were looking at these yesterday when we were thinking about introducing Fisher's geometric model, which I think you're going to hear about in more detail in a minute. But so we might imagine y and x. And if I draw this right, which is always the challenge because I'm not a very good artist, maybe if I drew it bigger. If I draw it bigger, then you'll see my mistakes bigger. It'll be better. So we'll call this x, y. And I want to draw it so that the fitness falls off more steeply in one direction than the other. So these are lines of sort of equal fitness. A contour map. It falls off much more steeply in y. Oh, it's not in any way. Not even an oval. It's really bad. Okay, pretend that it's really good. So if we are somewhere on this surface, here, and we ask ourselves, if all else being equal, how will natural selection change the trait, which is made up of both x and y? We can say that there are vectors given by b of x and b of y, which tells us the slope in each of these directions. And let's imagine that we have b of y this way. This is b of y. I'm sorry, beta of y. And a little bit longer for beta x. Okay. And so if they're completely, because we have the steepest difference. Okay, so if they're completely uncorrelated, if x and y are genetically uncorrelated, correlated at all, then it's very simple to think about what will happen, right? We just do vector addition, and we're moving to maximize fitness. It's wonderful. But basically what additive genetic correlation does is it makes it that you can't move, not always, you can't always move along the direction that maximizes fitness. So Fisher's fundamental theorem is not happening. Now, we're always going to, Fisher says this is always going to be positive. Why is it always going to be positive? Well, it's, positive or 0. Now, it'll be non-negative. Right? Because this is a variance, so it can be 0 or it can be positive. So we're always going to move. We're either going to not move at all, or we're going to move towards maximizing fitness on this surface. But if we have, let's say, that these are correlated, maybe, let's say, highly correlated. Now what can we do? Well, this says that if you increase x, the genetics are such that you increase y. If you decrease x, you decrease y. So you can move that way, increasing both, or you can move that way, decreasing both. But you can't move in this direction, and you can't move in that direction, because the genetic covariance doesn't let you. So what can you do? Well, that's the fast path. Instead, you can, if it's possible, you can take what's called the slow path. The slow path needs both x and y to both increase, or x and y to both decrease. So Many things are possible, but I'll show you a slow path. In color, maybe. My one use of color. If it's going to be my one use, I should use the pink. So we can imagine that we could take a step in this direction. Both are increasing. And maybe another step in this direction. Both are increasing. They're both still increasing. One's very, not so much as the other. I don't know. A whole bunch. The smaller steps would be better because if I get far enough, am I far enough yet? That can't happen. If I get far enough out, I can take steps that are negative, negative this way. And now maybe I can, get there in some reasonable amount of time. Something like that. So what happens? This is just a possibility. What's possible? What's possible is that it's possible to get in this very simple fitness landscape from here to here. But the way to get there is complicated, and it takes a long time. So what am I assuming by making this rather silly picture? That I think Kavita has sort of set the stage for, and other people have sort of pointed out. What's the assumption here? That R of A is not changing, and that landscape is not changing. But that seems not real. So what's probably going to happen is if this is indeed the slow path, that during some time in this path, there's going to be changes in the additive genetic covariance. So it may go from positive to negative, or all kinds of things can happen. And this surface may change. So why are we're especially interested in that because we're often interested in what happens to selection when the environment changes. So what happens to a population during environmental change? Well, environmental change changes the surface. It also changes these. It can even change the sign. So you are perhaps wandering towards, maximum fitness. That's where natural selection is taking you. But things change on the way there. So now, we could make a silly cartoon that there's a new thing, this goes down and something else goes up. And now you find yourself wandering in a different direction. So Daniel presented sort of a model of thinking about change let me not use the right term. He's in a high-dimensional phenotypic space, and the idea that if you introduce a correlation, a feedback between phenotype and environment, and he was thinking, I think, really of sort of microbial communities where the phenotype of the microbe is like releasing chemicals and changing its environment. So there's this feedback, and a result of that is this wandering on a surface. But what I'd like you to realize is you'll get that same behavior even if you do not have a direct correlation between environment and genotype, as long as you have non-constant fitness surfaces. And any kind of correlation. If there's any kind of correlation, you can't just climb to the top of that peak in the Alps that we were looking at as sort of the symbol of perfect fitness. You're going to wander around in these saddles, and you're going to wander around possibly always. I had a teacher who said, well, this is what makes evolution interesting, right? If correlation is 0, and you just go here and environment doesn't change, it'd be very unlikely that you would see all the diversity that you see around you, right? We're pretty sure that one of the things that's generating a lot of diversity is the fact that things are not constant, that things are correlated, and there's sort of complicated ways of moving through phenotypic space. So that was like a preaching moment. I was like convincing you of my beliefs. Oh, it's very early. Okay, well, I thought it was going to take a really long time, and it did not. So I'm more than happy to end a little early, but why don't we talk a little bit if you have some questions about this and just think about this in any way or not. Yes? 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 So one of the things, so if you're looking at a trait X, well, if you're not doing any experiments, you're going to be. But let's say you're looking at a trait in a population. Oh, our sparrows. I can go back to that. You saw this event, and we saw that the larger wings had higher fitness. It was correlated with higher fitness. And let's say you said, fine, I'm going to observe sparrows in Kansas for many years. I'm going to spend all my sad life watching this up. And you're measuring their wings, and you notice that the wings are not getting any bigger, right? Well, one possible explanation, in fact, a classic explanation, is that there's correlation with other traits that are under natural selection, and that trait has no ability to change independently. Even though selection is hammering it down, it can't change because it's tightly constrained. This is the idea of evolutionary constraint, and sometimes the term developmental canalization, right? So evolutionary constraint. Sometimes we observe traits for which there appears to be genetic variation, there appears to be selection, and yet the trait does not change in the direction that we think it should be. A classic example of that is, and sort of maybe a little obvious, Drosophila geneticists love bristles, bristle number. They love to breed for more bristles or fewer bristles. It's like, but can you select Drosophila for more bristles on one side and fewer bristles on the other, for asymmetry? So there have been a lot of experiments trying to select for asymmetry. There is variation for asymmetry. You see some Drosophila who are more asymmetric than others, so it seems to be some. And there's even some evidence that there might be some genetic variation in it. But it never responds to selection. And that's because probably a very strong genetic constraint. Because it's, well, in that case, it's probably very obvious. There's probably genes that set up the number of bristles you have, but there's probably not genes, the way insects develop, that set up the number of bristles you have on the left and the right side. Maybe the front and the back? Yes. Anterior, posterior, that works, but one-sided, doesn't really work. And this trait will not change. There'll be some variation. Asymmetry in general, organisms, asymmetry in most symmetric organisms. We are all, have you ever done the thing where they take your face and they show the other side of your face and how it looks, well, you may have a beautiful symmetric face, but my face looks extremely strange, which means that it's asymmetric. And that's usually developmental. So the asymmetries that we see in most organisms that are bilaterally symmetrical have to do with developmental accident. The programming, if you like, underneath is setting up symmetry. And then there's some asymmetry. But you can't really, it's very hard to select for asymmetry, which I was just thinking about the fish whose eyes crawl up onto the side of their head. So, but they start off symmetric, right? They start off symmetric. Later in development, they undergo a very strange sort of hormonal change that leads to one side or the other, not always the same side, becoming very asymmetric. So those are very interesting questions, I guess, for this idea of constraint, because symmetry seems to be a strong constraint in some organisms. But yeah, you can definitely see traits that do not respond to selection the way you think it would be. And if you think about this kind of situation, it makes sense that some traits won't change. But temporarily, right? If I change the environment and I change the genetic background, these things will change, and maybe in the future they will respond to selection. We can only sort of say what's happening right now with the organism the way it is. And remembering what Ophelia said, that the organism right now is a product of evolution. So it has depleted and generated genetic variation in various ways over time that we can't necessarily see, but is there. So another answer could be there's no genetic variation for the trait. Usually you can tell, though, if there's some genetic variation. Depends on the organism. Yes? Other questions? What's the fish with the eyes on the side of the head? Flounder? Is it flounder? No. Maybe. You're a biologist. It's a fish. It's not, I don't think it's flounder. Okay. So should you come to the board and do some problems? I'm happy to be done if there's no more questions.